Okay, thank you, G, for the introduction, and all the organizers for giving me this opportunity, and also making this technically possible. I hope it works well. The internet connection here in Connecticut was rather spotty for the last 12 hours, so please let me know if there is a problem. So I'm not going to talk about population genetics. I'm not going to talk about avian digits that G and I are working on together. We actually had a very nice symposium last year in China that G organized, but evolution of cancer malignancy. Let me see whether that works here. Okay, so before I get into further detail, I want to point out that evolutionary cancer biology is actually a field that consists of two rather independent sub-fields, rather orthogonal. One can be called the somatic evolution of cancer and analyzes cancer progression within a patient as an evolutionary process, which is of course appropriate because we all know that cancer cells undergo mutation and that the selection among tumor cells and so on. On the other hand, there is comparative cancer biology, and that is quite different because it looks at really evolved species differences that are relevant for species differences in cancer biology. And in there are also two directions that you can take. The one is to ask about cancer susceptibility differences between species, like for instance the question of Peter's paradox, which is basically the question why large mammals like blue whales and elephants don't get more cancer as you would expect based on the number of cell divisions they have to go through. And that's actually a reasonably active field of comparative cancer biology. Much less is done in the second area, namely the vulnerability to cancer metastasis. So how and why cancers turn malignant. And you have to realize that those are actually two different kinds of questions. The start of cancers is one thing, and then the cancer can either stay benign or can turn malignant. And those are biologically two different stages of an extended process. Just to make this very clear, here we have an example of an Indian cow that has a quite substantial melanocytoma, but otherwise then the cow is healthy, doesn't show any secondary tumors. So this cow certainly has a tumor, about 2 kilograms, but is entirely unaffected other than the weight that it's up here at his neck. Now, in contrast, if you look at humans, already 4 millimeter thick melanocytomas, reduce your survival rate to less than 50% on the 5-year scale. So there's clearly a quite different, quite substantial difference between species, let's say cows and humans, how the progression of the cancer is going on. Now, at this point, I have to make an argument that tells you that I didn't get into biology to do something about cancer, but it's actually a side effect of our recent year focus on the evolution of mammalian implantation, the female reproductive biology of mammals, particular placental mammals that have this invasive placentation versus marsupials like this opossum here has non-invasive placentation. We'll talk about it. And it is this connection that actually brought me eventually into cancer biology, because it turns out that this cows in general, that are less vulnerable to cancer malignancy are also having a form of placentation that is less invasive than the one that you would know, let's say, from humans. And of course, there's also a Viennese connection there, and knew for many decades about our Spanische Hofreitschule, the Spanish Hof Court Riding Academy, that the horses that they are using, the Lipizzanas, actually have a very high rate of developing skin cancer, like you can see here. About 50% of them, if I'm correctly informed, develop these primary lesions, but they very rarely are dying from it. So it's a quite similar situation to the one that I explained about the cow before. And as it happens to be, horses also have a superficial placentation. So this connection, this potential connection between cancer biology and placental biology, is the one that brought me into this field. Of course, we were not the first to suggest such a connection between tumor metastasis and embryo implantation. And I'm sorry for that. It's very dry here. And people have primarily pointed out that embryo implantation and tumor metastasis actually use quite similar cell biological and molecular mechanisms, and therefore one should expect a connection there to exist. For instance, the human embryo is invading the endometrium at the beginning of pregnancy, and cells are invading the endometrium in a quite similar way as metastatic tumors are invading the surrounding tissue when they start becoming malignant. So I have divided my talk into two parts. One is sort of on the comparative biology of placentation and malignancy rate. And if you don't care much about comparative biology, then just hang in, because in the second part, I'm going to talk about experimental work on the mechanistic biology of species differences in malignancy rate. So let's first start with the comparative biology of placentation and malignancy rate. The first question I would like to answer is the association between placenta type and malignancy rates actually systematic? Because so far I only have shown you anecdotes, one race of horses and one cow in India. Let's see whether that is a more robust connection. Before I do that, I have to introduce a little bit of terminology that maybe not all of you are familiar with. It is a classification of placenta types that actually goes back to the Viennese anatomist Grosser, who first classified different placentas from different species by the number of tissue layers that separates the fetal blood from the maternal blood. So when I say a placenta is epitheliochorial, what I mean is that the epithelium of the placenta is simply attaching to the epithelium of the uterus, and then you have the 1, 2, 3, 4, 5 tissue layers separating maternal and fetal bloodstreams. The next most more invasive form of placentation is one where the epithelium of the uterine endometrium is eroded, and the epithelium of the placenta is directly contacting the connective tissue and the blood vessels of the maternal side. What you find is in dogs and cats, and up here epitheliochorial you see cows and horses and pigs and so on. And the most invasive form of placentation is the hemochorial placentation, where not only the epithelium but also the part of the maternal blood vessels get destroyed, so that eventually maternal blood is in direct contact with the epithelium of the placenta. And that is the situation that we know from humans, rodents, and many other species. So the first question was, is there a connection between, can we substantiate a connection between placenta type and malignancy rates? And the best, most extensive data was actually this quite old paper from the National Cancer Institute, from data from over 200,000 animals. And I made an undergraduate of mine working on this, Alaric de Sousa, and we in fact found that for many categories of cancers that they have found in these domestic animals here, the cats and dogs that have the more invasive placentation than cows and horses, actually the malignancy rate is higher in cats and dogs. Not in all of them. There's one category of cancers where we didn't find a difference, but in many of them, skin cancers, glandular epithelial cancers, and connective tissue cancers, probably sarcomas, there was a quite strong difference between the species, and a difference that corresponds to invasiveness of the placenta. Now, more recently, there was additional data published by Amy Boddy using necropsy data from the San Diego Zoo. Well, my colleagues and I disagree with her interpretation, but taking the data that is in her paper, and combining it with our data that I just talked about, you get actually this result where epitheliocoral species have the lowest estimated malignancy rate, the highest in hemocoral, so highly invasive placenta is correlated with a higher rate of malignancy. So that is reassuring. It's still, the numbers are still sort of small because this comparative data is not readily available. There's not that much, and we certainly need more. But on the face value of it, it confirms this idea that there are species differences in malignancy rate that correlate roughly with the form of placentation. Now, in that data, there is one category here that Amy has correctly looked at separately, and that is the cancer rate or malignancy rate in marsupials. And that is actually interesting because if you look into textbooks, marsupials tend to be described as non-invasive placentation. So let's look into this a little bit in detail. So in general, marsupial placenta malignancy seems to be a counterexample because there's evidence of good rate of malignancies in marsupials, and the placenta type is described anatomically often as non-invasive. Now the problem is that let's say the opossum that we work on in the lab is we know most about. The so-called non-invasive placenta is actually different from what cows and horses are doing. So first of all, you have to understand that the opossum pregnancy is rather short. It's quite typical for many marsupials, about 2 weeks from fertilization or copulation to birth, gives rise to quite immature neonates. And here's a histological picture from a student of mine where you see the uterine epithelium is easily detached from the placental epithelium. However, what is not appreciated so broadly is that actually for about 12 days out of a 14-day pregnancy, the trophoblast, or the placenta, and the uterine epithelium is separated from each other by a shell coat. So you see here this extracellular material here, and only during the day 12, the shell coat disappears, and there is a direct connection between, a direct contact between the placenta and the uterine epithelium that starts to do some crazy things that I don't understand really. But anyway, the main point here is that of a 14-day pregnancy, there is about 12 to 13 days where the attachment just doesn't happen. And once attachment is happening, within 1 to 2 days, birth is actually precipitated. If you compare that with a mouse pregnancy that takes 19 to 21 days, the attachment and implantation happens after 4 days. And during this time here, whatever it is, 15 days to 17 days, the mouse placenta is deeply embedded in the epithelium of the endometrium. So the opossum pregnancy is non-invasive, technically epitheliocoral, because it doesn't have enough time to actually invade, right? There are only 2 days around here. And actually, if you look at the very end of pregnancy, there are signs that the placenta itself is actually invasive. It's at several points are breaching the uterine epithelium, but this doesn't lead to sustained invasive fetal-maternal interface, because once these things happen, inflammation is going out of hand and birth is precipitated. So this result here is entirely consistent with the overall picture that this is sort of an invasive placentation that just doesn't get invasive because it doesn't have the time to do it. Anyway, so then the question is, how did and when in evolution did invasive placentation evolve? Now the current consensus is that the placental mammals that you see here have phylogeny of it, where black is hemocoral, most invasive, and the light gray is epitheliocoral. The maximum likelihood estimates are that for 75% posterior probability, the ancestor of placental mammals probably had the most invasive form of placentation, namely hemocoral. Yet cows have a superficial placentation. So when did this happen? It turns out that the non-invasive placentation among placental mammals is actually a thing that evolved much later, secondarily. First we evolved an invasive placentation and then a non-invasive placentation. So the invasive placentation that we see in humans is actually relatively primitive compared to what we see in cows and horses and so on, because they have a derived form of placentation that is special. So what we observe here is that only those mammals which secondarily evolved non-invasive placentation, like the hoofed animals here, are protected from malignancy. It's not the marsupials that never were invasive are protected from it. So that supports the idea that maybe evolution of non-invasive placentation and evolution of less vulnerability to malignancy might be connected. So then the next question is, how come implantation is non-invasive in hoofed animals? What is the cell biology behind it? Is it that their fetuses or placentae are less aggressive, or is it that the mother found ways to defend herself against the invasion by the fetus? And there's some very old papers and experiments that, of course, need to be eventually repeated. And I'm working on a friend of mine who has access to large animals in his research to repeat these experiments where they have artificially produced ectopic pregnancies in the pig, which has a non-invasive placenta, and actually shown that an ectopic site, so outside the uterus, the placenta is still aggressively invasive. So their conclusion was, and this is one that we can also support with some of the experimental data I will talk about later, is that evidence on an inherent invasive property of the trophoblast is supported and shows that normal development in the pig is regulated by the uterine environment, saying that the cow and the pig and so on, the non-invasiveness comes from the fact that the mother is able to prevent invasion rather than the fetus being less aggressive. So let us summarize the comparative biology of cancer and malignancy that I wanted you to know before we go to the experimental work. So some mammals secondarily evolve non-invasive placentation, those are the cows, horses, pigs, and those animals, and others as well, but they are not as easy to work with, like tarsiers and so. These animals also happen to be less vulnerable to malignant cancer. So the non-invasive placentation is associated with non-invasive cancer. Non-invasive placentation is due to resistance of the mother against embryo invasion. So the hypothesis is maybe that the evolution of resistance to cancer malignancy is a side effect of maternal evolution for non-invasive placentation. Okay. So now I'm going over to the second part of my talk, where I report a little bit about our experimental work. And to set this up, we have to ask ourselves what could be the biological mechanistic reasons why some species have lower malignancy rate than other species. So one factor we should consider is that maybe species differ in the nature of the oncogenic mutations, which is possible to think about because the genomes are different. But overall, I think that's rather unlikely because all mammals and in fact all multicellular organisms like plants and all other animals, invertebrates, do get cancer. And the common cancer-driving mutations are on genes that are widely shared among mammals. So I don't think that this is a very likely direction. The other possibility, which is inspired by this work on the non-invasiveness of the pig placenta, is that there might be species differences in the way the stroma, meaning the cells or the tissue surrounding the tumor, reacts to the tumor signals. And there can be sort of two things to look at. One could be the interaction between tumor and immune cells. There's a lot of research going on in that direction. Or it can be also the species differences in which between the way fibroblasts, so the connective tissue cells surrounding the tumor, are interacting with each other. So it's actually this direction that we have pursued in our research, and that's supported or also inspired by the fact that the tumor, in most tumors, except maybe brain tumors, most tumors kill the patient not because of their own activity, but because they became malignant through the help of normal non-tumorous cells surrounding the tumor. Now, these cells are called cancer-associated fibroblasts, and they get created or get recruited into this role from, fibroblasts, mesenchymal stem cells, endothelial cells, which of course are also mesenchymal cells, under the influence of signals from the tumor to assume a phenotype which actually then helps the tumor survive and spread and so on, and even metastasis, so the spreading of tumors to other parts of the body, is usually not by single or tumor cells, but actually clusters of tumor cells with cancer-associated fibroblasts. So malignancy as a process is an interaction between the connective tissue surrounding the the tumor and the tumor cells themselves. So the idea, which we call evolved levels of invasibility, is the hypothesis that maybe species differ in their readiness with which they can get influenced by the tumor to help the tumor in its nefarious pursuits. So this is our hypothesis that we will start testing. So the first test that we did was published recently in 2019, and that's an essential collaboration with my colleagues Chititz, who only goes by one name, and Andrei Levchenko, who is actually the director of our Yale Systems Biology Institute. And what we did there was the following. We used some techniques that so Andrei Levchenko is a biomedical engineer, and he developed these culturing systems where cells are cultured on nano ridges, which has the advantage that cells that are migratory and lying on these ridges will preferentially migrate along those ridges. Basically, the ridges are simulating the presence of collagen fibers, which we all know cells use to direct their migratory direction. And the experiment itself is that we take trophoblast or cancer cells, label them with one or the other fluorescent dye, and co-culture them side by side with fibroblasts, and then we observe how quickly these invasive cells can actually enter a lawn of fibroblasts on the other side. So the fibroblasts are usually not labeled, therefore we don't see them here. And here we have a result here. I should move this. Okay, so what you see here is green labeled are cow trophoblast cells that are opposed by unlabeled human endometrial stromal fibroblasts. So you can see, even though the cow placenta in vivo is non-invasive, if they are faced with human fibroblasts, which are highly invasible, they actually invade quite rapidly. But that confirms, also the conclusion of this ectopic pregnancy experiment from 1971. And here now you see the case, the same cow trophoblast cells, but facing off with cow endometrial fibroblasts. And you can see that they're still trying to do, and they gain some ground, but overall their progress is frustrated by the cow endometrial fibroblasts that are here. So this in vitro result reproduces the in vivo difference, where when of course cow placenta is living in a cow uterus, these trophoblast cells, which do have an invasive tendency, get frustrated or contained by their cow fibroblasts. So that supports the notion that invasiveness is to a large extent a species difference between invasiveness are to a large extent determined by the biological nature of the fibroblasts, the stroma surrounding either the, in this case, the cells. And the same pattern is also found if we use, instead of endometrial fibroblasts, and use skin fibroblasts, and we have different cancer lines trying to invade. So skin fibroblasts, dark human skin fibroblasts, and gray cow fibroblasts, and you see that in all instances the human skin fibroblasts get more invaded by the cancer cell line than the bovine skin fibroblasts. The largest difference is here. This is a particularly malignant form of cancer cell line. So not only does the do the differences in the endometrial fibroblast biology determine the the degree to which placental cells can invade. The same pattern qualitatively you also see with respect to skin fibroblasts as they are facing off with cancer cells. Even more importantly, if we look at gene expression differences between human and cow cells and select those genes that are less expressed in cow cells and take human cells, endometrial fibroblasts or skin fibroblasts, and reduce the expression level of this particular gene in the human cell so that it mimics the expression level of the cow, the human cells also become less invasible. So here's extent of invasion. This is a TGF-beta1 knockdown, and this lower expression of TGF-beta1, they are also less invasible, and about half of the genes that we have tested are having this effect. There's some correlation with skin fibroblasts and so on. So not only can we say that the biology of the fibroblast determines how invasible, how much they resist invasion, we can actually manipulate the highly invasible human cells so that they become less invasible by trying to make their gene expression profile more similar to that of a cow. So this is what we have at this point. So fibroblasts account for at least part of the human bovine difference in invasibility, both in the endometrium as well as in the skin. And changing gene expression in human fibroblasts can make them more resistant to cancer cell invasion and trophoblast invasions, placenta invasion. But of course, one thing, these genes here, of course, were selected based on gene expression differences, but also because they are already known to be important in cancer and malignancy, right? So that was not an unbiased sample. So actually, we don't learn much more about the mechanisms of cancer invasiveness than we already know. So what we really want to know is to find an unbiased approach to identify mechanisms that cause species difference in malignancy rate. And this is what we did since this paper. And we approached this by culturing skin and endometrial fibroblasts from about 10 species and have one clade that has these hoofed animals as well as the carnivores, and then the primates and the rodents here that have the more invasive form, and this is the less invasive form, and then ask, what differences exist between them. So one example, one result from this data is if we compare evolutionary changes in gene expression in the endometrial fibroblasts and independently in the skin fibroblasts and see whether they change in concert, we find actually a quite high correlation. And we, of course, do this with phylogenetic independent contrasts to avoid, phylogenetic artifacts here. So that's probably the evolutionary reason why skin fibroblast biology and endometrial fibroblast biology tends to be correlated because they have a tendency of changing gene expression profiles in a correlated fashion. Here is one example, an extreme example, nice example, activin receptor 1. And if you use Hochberg statistical techniques, you can estimate that about 80% of genes have correlated evolution of the 8,000 whatever genes that have one-to-one autologs in our species sample. Okay, so if you go back to the phylogeny of all of the placental mammals, our species sample comes from these two, from the Archaeontoclerus and the Laurasia, and we compare these to these, and we would like to know what happened here that led to the less invasibility in these animals. So one observation that you can make if you look into the gene expression of hemo and endochorial species compared to the epitheliochorial. And then up here are the genes that have a lower expression in the more resistant ESFs, and down here the genes expressed higher in more resistant endometrial stromal fibroblasts. And what we see is that there are many more genes that are less expressed in species that are less invasible. So here you have an example. So with high TPMs, we have the human samples that have a high invasibility, and the low expression has a low, the species with low expression have a low invasibility. And here's another example going in the other direction. So that means resistance to invasion is more often associated with lower gene expression of a particular subset of genes, namely those genes that show a correlation with invasibility in general, and that's a pattern that we will see further. So then the question is, so what are actually the genes that are lower expressed down here? We used the Ornstein-Uhlenbeck model to select or filter out the genes that have this tendency, and this is work done by Anna Suya-Dige, a computational biologist. And by the way, Ornstein-Uhlenbeck model is of course a stochastic model that's a long time, but Thomas Hansen was the one who pioneered the use of this model in comparative biology. So what you can do is you can estimate the parameters of a model that assumes that there's only one sort of central tendency for gene expression, so evolution of gene expression changes, but it's always sort of tending towards one optimum, one preferred expression level, or you can run a model with two optima, one for the Laurasia and one for the IR ontocleris, and we select then those genes where this model is much better than this model, and then calculates the difference in these optimal values. And the way we calculate it is that if this is positive, then the expression in the Laurasia is lower. Okay, so one thing is that these delta values, of course, is calculated for the skin fibroblasts and the endometrial fibroblasts, and you also see here a positive correlation in this delta theta, similar to this correlated gene expression that we found using phylogenetic independent contrasts. Okay, so you then take the genes that have this Laurasia low characteristic and ask them, what are the functional gene ontology categories, and then you get a whole shitload of different things, from signaling to cell motility, hematopoietic protein, all kinds of stuff. So it's really hard to make any rhyme or reason out of this collection of gene ontology terms, until you actually, I don't know whether I can move this windows here away, but can you see this up there in the right upper corner? Anyway, so this is a list of these Go categories, and the highest enriched category is this regulation of amino acid metabolic processes with very low P and false discovery rates, and there are 13 genes. And then if you look into this the gene set, there are some that don't make a whole lot of sense, and then there's a huge number of them that all have to do with the proteasome. So the proteasome is of course this machinery that breaks down proteins in the cell, and that goes on. So if you take, let's say, Wnt signaling, which is one of these Go categories here, some of them, but the majority are the same genes that have to do with the proteasome machinery. And here's another Go category, NF-kappa B, all of the ones that are identified as belonging to this signaling pathway are actually the proteasome genes. And we can also get the same result if we use David-Keck pathway annotation. The highest enrichment you find in proteasome. So that's, of course, sort of a head scratcher, because invasiveness and proteasome have not been connected yet. A few words here. Here you have a 3D model of a proteasome, which is of course a multi-protein complex, and it has sort of two parts: a 19S regulatory particle, that is the red thing here, and then a 20S core particle, that is in particular here, this blue part, consists of the enzymes that actually break down the proteins that get into this machinery here. Well, this upper part, the 19S regulatory protein particle, is one that regulates which kind of proteins get actually invited into the proteasome and degraded in here. Now, if we look into this proteasome enrichment here, we have 12 genes there, and it turns out that 66% of these 12 here, of course 8, are from the regulatory 19S particle. Only 33 belong to the core particle. So they the most of the genes that are have a lower expression in the low ratio area and are enriched are actually genes that make proteins for the regulatory particle that determines which kind of peptides get admitted into the proteasome. Here's an example of the gene expression profile across species. For one of them, PSMD3, red are the expression levels for the endometrial and blue for the skin fibroblasts. Here you have the species names, and here the phylogenetically reconstructed ancestral levels. Sorry, here it should be blue. And if you just look at the average, the Eurasia of the area have about 100 TPM less than the arc ontoglirus, and also in the skin fibroblast there's over 100 TPM difference on average. So of course, you scratch your head and ask, well, proteasome is nice, but does it have anything to do with invasibility? So we turned again to our invasibility test that was done by Chititz in his lab at UConn. And we chose 3 of those genes, PSMB5, D3, and D13, and all of them, when knocked down, decrease the invasibility of human skin fibroblasts. So that is consistent with the idea that this correlation between lower expression of these genes in this species and the correlation with lower invasibility may actually be causal, because we can produce the same effect in human skin fibroblasts by decreasing the expression of those genes. Okay, so but still, it's still not clear how and why proteasomes should relate to invasibility. And here I'm returning to this model that I showed you earlier, namely the fact that malignancy invasibility, at least in cancer cases, requires the cooperation of these cancer-associated fibroblasts, and they get recruited through signals from the tumor cells. And so it's also important to remember, your cell biology 101, that many signaling pathways require proteasome activity for the activation, for instance, by breaking down some inhibitory proteins or similar things. So the hypothesis is that lower proteasome activity may attenuate the tumor fibroblast communication, meaning that if the proteasome is less active, the signals from the tumor may have less an impact on the behavior of the fibroblasts. So I call this the Odysseus pact hypothesis, where, as you remember, at one point the crew of Odysseus' ship were faced with the distractions of the sirens, and they decided to proceed by simply putting wax into their ears and just not listen to the siren calls and just go on steering their ship through the troubled waters. And I think the similar way of thinking could be applied here, that the fibroblasts are refusing to hear or being influenced by the siren calls of the cancer cells. So that's a nice story, but can we test this? So one test that we did just recently, and that is preliminary data, but it's so exciting that I cannot help but tell you about it. That was done by Cody Limber, as a graduate student, did a rotation in my lab, and the prediction was that if we knock down some of these proteasome genes, that will attenuate the response to some signal, and one of the signals that it shows is tumor necrosis factor TNF. So we had these 5 treatments. One is just non-treatment control, vehicle control for everything. Then one is 1 hour of TNF treatment. The other 3 are knockdowns of one of these proteasome genes, and then treating with TNF, and then have as a readout RNA-seq. Here you see the ordination, so untreated, TNF treated, and knockdown TNF treated samples here, so they separate in this direction. And what I then did was I looked at what are the most induced genes under TNF treatment, so that are at least 10 times induced and that have at least a 10 TPM expression after TNF treatment. And there are 22 of them, one of which is annotated as a pseudogene, so I threw it out. But you get what you expect here, all of these inflammatory mediators that you would expect in response to TNF. So that's fine. And then I asked, what is the effect of the proteasome knockdown on these highly induced genes? And it turns out that the majority of them, if you look at the knockdown effect in a relative difference, the majority of them actually have a negative effect on the response to TNF. So you have here blue is the effect of B5 knockdown, D3 knockdown is this reddish thing, and gray is the knockdown effect of PSMD13. If you look at the genes that have a negative effect for the knockdown, PSMD3 has 18 genes that get negatively affected in their expression, and the binomial test is highly significant. Here you see the knockdown efficiency, so that these differences are not easily explained by different knockdown efficiencies. And also, if you looked at the actual magnitude of the changes, the magnitude changes in the PSMD3 knockdowns are much larger, from over 40% to whatever 20 or so percent in the most affected genes. Here you see the same data and in actual expression data. I already threw out the B5 knockdown because it was uninteresting. So here now the D3 knockdowns are the gray ones. Red is the TNF effect from non-expressed to expressed, and you can see that it has a quite substantial consistent difference here. So far the most causally probably most important gene is probably PSMD3, which produces a protein that's called RPN3 here in the regulatory lid, and it's plausible that this protein plays a role in specifically process the specific proteins that are necessary for the activation of signaling pathways. So, of course, a lot of work still needs to be done, but what I can say is that this hypothesis of the Odysseus pact among fibroblasts has survived one falsification attempt. We will do more experiments as I'm speaking here, but I think it is an interesting novel direction and actually explores novel molecular biology of cancer invasiveness that hasn't played a role in the, as far as I know the literature, has not played a role in biomedical cancer biology research. So it just shows that using comparative biology can actually help you discover new forms of biology, of mechanistic biology, and not only telling stories about species differences. So the summary is that malignancy rate and placental invasiveness tends to be correlated. Animals with less invasive placenta are less vulnerable to malignancy on average. Lower malignancy rates are in part explained by resistance of tissue fibroblasts, the stromal cells. And one factor contributing to lower invasibility seems to be a lower responsiveness to signals from the tumor that can be mediated by various mechanisms, one of which, the one that we chose to look more closely, was the one had to do with proteasome degradation. And with that, I thank you for your attention, and I'm even less than 60 minutes at the end, and acknowledge my collaborators in the lab, as well as my collaborators outside my lab. And with that, I will shut up and thank you very much.